Good morning, everyone. Welcome to the UCI School of Medicine Dean's Distinguished Lecture. I'm Dr. Michael Stamus, Professor of Surgery and Dean of the School of Medicine. I'm so delighted you are here for this special event. I want you to note that it's called Dean's Distinguished Lecture with the apostrophe before the S, but we should really move that apostrophe and it really should be Deans plural because the Vice Deans actually select, arrange, and help me host the lectures. You will hear from Vice Dean Jeff Abbott shortly. We are dedicated to advancing medical knowledge and clinical practice through scholarly research, physician and other healthcare givers, education, and high-quality care, which embodies our tagline, Discover, Teach, Heal. We are ranked as one of the top 50 U.S. medical schools for research by U.S. News and World Report, and we nurture the development of medical students, resident physicians, numerous other trainees and scholars in the clinical and basic sciences, and support the dissemination of research advances for the benefit of society. Each year, we educate more than 500 medical and graduate PhD students and train more than 700 residents and fellows at the UCI Health sites and affiliated institutions. Over 900 full-time faculty members are involved in teaching, providing cutting-edge medical care, and conducting research into the health challenges facing the 21st century, including, of course, the pandemic. The school has 24 departments ranging from basic science research to clinical, medical, and surgical specialties, including, of course, ophthalmology. I'd now like to introduce my colleague, Jeff Abbott, Professor of Physiology and Biophysics, Vice Dean for Basic Science Research, and Senior Associate Dean for Academic Personnel, to share a few words about the Dean's Distinguished Lecture Series.
Thank you, Dean Stamos. I'd also like to thank everybody in the Dean's office who helped put this together, including Rachel Correll, who's with us this morning, helping out as always. So it's my distinct pleasure to be here today and give a few words about the Dean's Distinguished Lecture Series. It was established in 2019, and it was designed to bring together influential leaders from healthcare and academia to share their insights, experience, and anecdotal stories with our faculty, staff, students, and community members. The program features a series of speakers representing innovation in basic sciences, advances in clinical medicine, and developments in clinical research. This fall, we have partnered with the Department of Ophthalmology and the Center for Translational Vision Research to bring you a very special virtual distinguished lecture featuring a Nobel laureate. While we wish we could be together in person, we're extremely proud to be able to host this event safely via Zoom. Please allow me to introduce Baruch Cooperman, the Roger F. Steinert Professor and Chair of the Department of Ophthalmology, Director of the Gavin Herbert Eye Institute, and Co-Director of the Center for Translational Vision Research, to share a bit about the Center for Translational Vision Research.
Thank you, Vice Dean Abbott, and also, of course, to Dean Stamos. We are so delighted to partner on this event and to elevate our ongoing Center for Translational Vision Research Virtual Ophthalmology Research Seminar Distinguished Speaker Series. The Center for Translation Vision Research is focused on fostering vision research at the Gavin Herbert Eye Institute. The purpose of the center is to increase the knowledge of normal vision and blinding disorders and to create innovative technologies, solutions, and educational programs that improve visual health. To achieve the center's mission, scientists, engineers, and physicians devote their time and talents to discoveries in the basic and translational sciences, forming the bedrock of inventive therapies for the prevention of or treatment of visual diseases. The resultant breakthroughs in basic vision research will lead to new therapies for patients suffering from or progressing towards blindness. And now, please allow me to introduce Professor Chris Pelczewski. Chris is a distinguished professor and the Irving H. Leopold Endowed Chair in the Department of Ophthalmology. Chris is also a Donald Bren Professor, the highest honor that we can give to some of the faculty member at UC Irvine, and importantly is the founder and co-director of the Center for Translational Vision Research. Chris will present this morning's distinguished speaker.
Thank you, Dr. Cooperman. Good morning, everyone. Today, I'm really thrilled to introduce you our distinguished speaker, a truly remarkable man. Nobel laureate Dr. Robert J. Lefkowitz is James Duke Distinguished Professor of Medicine, Biochemistry, Pathology, and Chemistry at the Duke University School of Medicine. He is associate of the Duke Initiative for Science and Society and a member of the Duke Cancer Institute. Raised in New York, Bronx, in family of Polish heritage, Dr. Lefkowitz is married and has 5 children. He trained to become an MD at Columbia University. In 1968, he began to research career at the NIH as a clinical associate. Upon completion of the fellowship at the Massachusetts General Hospital in 1973, he was recruited by Duke University Medical School to join their faculty to develop a program in molecular cardiology. He started appointment at Duke University in rank of associate professor in medicine. He has been investigator of the Howard Hughes Medical Institute since 1976 and began his research career in the late 60s and early 70s, when there was no clear consensus what specific receptor for drugs and hormones even existed. This was truly a very exciting time in receptor pharmacology, kind of like a quantum chemistry on the beginning of 20th century. Much of the research effort for a number of receptor systems in early 70s related to growing realization that the receptor structures responsible for stimulating cyclic AMP formation involve separate proteins. For agonist recognition, those are receptors, cyclic AMP synthesis, those are adenylate cyclases, and the transducers of information from the receptors to adenylate cyclase, and those are heterotrimeric G protein. This work led to a concept of GPCR, of the vital role of those G protein in signal transduction processes. This largely arose from the considerable effort of the groups of the 2 other Nobel laureates, Martin Rothbell from North Carolina and Alfred Gilman from Dallas, Texas. In visual system, rhodopsin was linked to G protein by Lubert-Stryer. Dr. Lefkowitz won the Nobel Prize in Chemistry in 2012 for study of this G protein couple receptors. So very quickly, communication between cells in our body are managed by substances called hormones. Each cell has a small receiver known as the receptor, which is able to receive hormones. In order to track those receptors, in 1968, Dr. Lefkowitz attached radioactive isotope iodine to hormone adrenaline. By tracking the radioactive emitter by the isotope, he succeeded in finding the receptor for adrenaline and to study how it's function. This work culminated in collaboration with Merck scientist Katrin Strader on hallmark paper in Nature in 1986, revealing the nucleotide sequence and virtually proving the existence of this receptor. Remarkably, the new receptors, beta-adrenergic receptors, structurally resemble rhodopsin, which has been sequenced few years earlier, without doubt the best studied GPCR. It was later discovered that there is an entire family of receptors, around 600 we know today, that exist and acts in a very similar way. Those are those G protein couple receptors. Approximately half of all medication used today make use of this kind of receptors. Dr. Lefkowitz has been elected to a membership of the National Academy of Sciences, National Academy of Medicine, the American Academy of Art and Sciences, and American Society for Clinical Investigation, as well as Association for American Physicians. Along with the way of receipt of Nobel Prize, he received a number of other awards for his research, which are detailed in his biography on the distinguished lecture list of the Dean website, and his book. And again, I wanted to encourage you all to read this remarkable story of a man who is truly a pioneer in modern pharmacology. So without further delay, please join me in welcoming Dr. Robert J. Lefkowitz as our distinguished lecturer to speak on the topic, A Tale of Two Callings.
Thank you very much indeed, Chris, and everyone else for those wonderful introductions. I think we all have a sense of what we mean by the word calling. It's a very deep-seated sense that you were destined to do a particular line of work. And I've been remarkably blessed in my career to have experienced this sense of a calling on two separate occasions. Initially, as a very young person, to the practice of medicine, then later on to the practice of science and research. So what I wanted to do today is tell you a little bit about my story and how my career evolved, and stress some of the lessons that I've learned along the way that might be perhaps of use to you. So the first calling I experienced was to the practice of medicine, and I attribute that to this man, Dr. Joseph Feibush, who was my family physician in the 1940s and 50s when I was growing up in the Bronx. So he would make house calls, come to the house when I or a member of the family were ill, toting that black bag of his, out of which he would produce all manner of magical instruments, from a stethoscope to an otoscope to an ophthalmoscope or his prescription pad. He'd let me play with these different things, especially his stethoscope, and listen to my heart. And so I would say by the age of 6, 7, 8 years old, as shown here, I was entirely convinced that I had no goal in life other than to be a practicing physician, or perhaps center fielder for the New York Yankees. But I think I was focused on the idea that someday I would grow up to be just like Dr. Feibush. And I focused on this goal unwaveringly. When it was time to go to high school, I was admitted to the Bronx High School of Science, which was a remarkable public, I stress public high school, that was open to gifted and talented students in New York City who could score high enough on an entrance examination. From there, it was on to Columbia College, where I was a chemistry major, and then on to Columbia Medical School, shown here at my graduation in 1966 with Mom and Dad. My father was to die 2 years after this photograph was taken, at the relatively young age of 63, of his fourth heart attack. My mother also had a heart attack at age 56. So genetically, I knew I was marked early on with premature coronary artery disease coming at me from both sides of my family tree. And in fact, in 1994, at age 51, I developed angina and underwent quadruple bypass surgery. But here I am, some 27 years later, due to wonderful advances in cardiovascular medicine and surgery. I want to stress that to this point in my career, I had never done any research, nor had I desired to do so. I was entirely focused on the issue of becoming a practicing physician. So, for example, when in medical school, opportunities arose to do research electives, I invariably didn't take them and instead did clinical electives so that I could hone my clinical skills as rapidly as possible. I stayed on at Columbia, shown here as a medical intern, and did my medical internship and 1 year of residency in 1966 and 1967, and would have gladly stayed on to finish my clinical training and go into full-time practice were it not for a cataclysmic event, the Vietnam War. This was a very unpopular war. There were demonstrations, even riots, and none of us, or at least none of the people that I associated with, wanted to support it. We generally felt that the war was at best immoral or at worst illegal, and we would have done anything to avoid supporting it. Alas, there were very few things that one could do at that time to avoid the doctor draft. Now, at that time, during the Vietnam War, there was a lottery draft for all men over 18. But for all physicians, there was no lottery. Everybody was drafted upon graduation from medical school, and you went into either the Navy, the Army, the Air Force, or the United States Public Health Service. Now, if you were in any of the first three of those services, you could pretty much bet that you would spend one of your 2 conscripted years in Vietnam. The only way to avoid that fate was to be drafted into the United States Public Health Service, because many of their assignments were in the United States, including some very plum assignments at research establishments such as the CDC and the NIH. So, as you can imagine, it was extraordinarily competitive to get assignments to the United States Public Health Service. But fortunately, I had high grades, high standing academically, and so I was able to get a commission in the United States Public Health Service and be assigned to the NIH, which I was very pleased about. We did not wear uniforms the way this officer is shown. As I'll show you momentarily, we wore white coats. As a result, we were relatively unfamiliar with protocols about uniforms, salutes, etc. But we were aware that if you were active duty military and in uniform, you could fly free, standby, anywhere in the United States. So groups of 6 or 8 of us would purchase one uniform, and whoever happened to want to fly would wear that uniform. As you can imagine, in any such group, the heights might have ranged from 5'5" to 6'5", so it made for some pretty uncomfortable looking appearances. I was fortunate in that at 6 feet, I was pretty much right in the middle, and so the uniform actually fit me pretty well the 2 times that I wore it. But people would get into mishaps. I had one colleague who wore the uniform to the National Airport, was waiting for his flight when he perceived what he took to be a senior officer coming to the same gate. Knowing from a handbook that we all read when we first started that it was the responsibility of any officer in uniform to immediately initiate a salute to a more senior officer, and seeing all the seeming decorations and epaulettes and stripes on the officer, he jumped up, snapped off his best imitation of a crisp salute, and which the senior man returned. And then the senior guy asked my colleague, what branch of the service are you in? And he said, I'm in the United States Public Health Service, sir. What about you? And he said, well, I'm not in any service at all. He says, I'm the pilot of the plane. I'm just here waiting for the flight, just like you. So that tells you how much we knew about military protocol. Now, the program that I was involved in, that is United States Public Health Service officer, we were lieutenant commanders, assigned to the NIH, were officially referred to as the ATP, the Associate Training Program. But the more common moniker that was applied to this program was the Yellow Berets. This was obviously a pejorative moniker based sarcastically on a perversion of Green Berets, which of course were our most elite combat troops. Yellow, of course, implying cowardice. But as I said, for most of us, this was not an act of cowardice, but rather an act of not wanting to support the war. In any case, we actually kind of liked this sobriquet and came to call ourselves Yellow Berets, which came to be sort of a badge of honor for reasons I'll show you momentarily. We split our time between caring for patients and learning how to do research in the laboratory of skilled mentors. I generally had a typical division of labor with about 80% of my time spent in the laboratory and about 20% of my time taking care of patients on the services who were hospitalized at the clinical center at the NIH to take part in clinical research studies. The legacy of this Yellow Beret program was nothing short of remarkable. Just a couple of slides to highlight that. In 1998, you can see that a very substantial percentage of professors of medicine, including at my own institution, were individuals who had previously been Yellow Berets. This slide is out of date by about 15 years, but even 15 years ago, you can see that the numbers elected to the National Academy of Medicine, the Institute of Medicine, now known as the National Academy of Medicine, and other high positions, was really quite remarkable. But perhaps the single most remarkable statistic is shown here. So this was a small program, maybe 50 to 100 individuals per year sent to the NIH as public health service officers. And we were all physicians, and with one or two notable exceptions, none of us had any prior research experience. We were basically all just young physicians out of medical school. And here we were. Nonetheless, between 64 and 72, the peak years of the Vietnam War, 10 of us, shown in this yellow stripe and in blue rectangles, 10 of us would go on to win the Nobel Prize in either medicine or chemistry. As you can see, for 4 of us, now up here, let me just say, up here were in the first rows our mentors, and above them, their mentors, and above them, their mentors, etc. So it's basically a family tree. And the legacies are remarkable. You can see that 4 of the 10 of us trained with a Nobel laureate. Anything colored blue is a Nobel laureate. But for the other 6 of us, you don't have to go back any further than a scientific grandparent to find a Nobel laureate. And if you look at some of these lineages, they're studded with laureates. Look at some of these, where you'll have 6 or 7 generations with, in this case, only one individual who is not a Nobel laureate, and that's the legendary Earl Statman, who in turn trained 2 Nobel laureates. So obviously, and I'll talk more about mentoring in just a few minutes, I think this speaks to the issue of the importance of mentoring and the fact that there are subtle things about approaches to science that can be passed from mentor to mentee. Another interesting aspect of this family tree is that if you go back 4 or 5 generations, you find a number of Nobel Prize winning physicists and physical chemists, even though, of course, all of us were biomedical scientists. So you find people like James Chadwick, Erwin Schrödinger, Niels Bohr, Linus Pauling, etc. So obviously a lot of cross-fertilization. This photograph shows me at the NIH in 1968. And it was here at the NIH that I had my first research experience. It was also here at the NIH that I experienced something that I had never experienced before in my life. Unmitigated, protracted failure. I had never experienced prolonged failure at anything that I attempted in my life or career, either academically or even in sports where I was not gifted, but found that I could generally rise to the middle of the pack at least by dint of hard effort. But here, no matter how hard I worked, no matter how I focused on the problem at hand, I seemed to make no headway. And so this just convinced me further that research was certainly not anything that would play a role in my future career. And so at the end of the first year of all this failure, I made arrangements that at the end of my second year, I would move on to Boston and finish my clinical training at the Massachusetts General Hospital. Now, as luck would have it, during my final 6 months at the NIH, I finally made some headway. I met with a little bit of success, was able to publish my first few scientific papers, and I was really beginning to enjoy it. But I still was not as convinced as my mentors were that this was something for me. And despite their remonstrations that I should remain at the NIH for additional period of years to follow up on the leads that I was developing, I decided that, well, I had really had enough, and off I went to Boston. This is a photograph of me at the Massachusetts General Hospital in the fall of 1970 as a young medical resident. And there I threw myself into the care of patients, intense clinical work, which I had always loved and frankly been pretty good at. And that went on for about 6 months. And then during the holiday break, at the end of that first 6 months, back on full-time clinical service, I found myself preoccupied with the fact that, despite the fact that I was enjoying the clinical work, something seemed missing. I didn't feel completely fulfilled. And I eventually realized that what was missing was the lab. I actually missed the lab. I missed the day-to-day challenge and excitement of confronting research problems, etc. And so it was then that I really had my epiphany and realized that I was experiencing a calling to actually incorporate research at some level into my career, although I still obviously didn't know how much that would be. And so I found myself another mentor at the MGH, and over the next 2.5 years, split my time between finishing my clinical training and trying to make initial progress on another research problem. And then in 1973, I received a very nice offer to join the faculty at Duke University, which I accepted. I went down there and split my time initially between being a clinician, teaching medical students and residents on rounds, and also starting up my own laboratory program. I would say during the first year or two, I probably spent about 40% of my time doing clinical work and about 60% of my time getting the lab program up and running. But very quickly, as we started to make progress in the laboratory, I gravitated more and more to the bench. And I would say within 3 years, I was probably spending 85% of my time in the laboratory and only about 15% in the clinics. And that blend or division of labor persisted throughout most of my career. But then about 15 years ago, I hung up my stethoscope for good. And now I pretty much spend all of my time in the laboratory and in teaching. I'm not going to speak about my research in this talk at all. Chris gave a wonderful summary of the early years of the field and how it evolved and our involvement in that. I want to stress that when I began, as Chris said, more than 50 years ago, there was really no consensus amongst either biochemists or pharmacologists or endocrinologists that there was any such thing as a specific receptor molecule. I think the context of our early work is shown very nicely in this slide taken from Earl Sutherland's Nobel lecture in 1971. Sutherland received the prize for discovering the enzyme adenylate cyclase, which catalyzes the formation of cyclic AMP, the so-called second messenger of hormone action. And as you can see, he knew that hormones like adrenaline and glucagon could stimulate this enzyme. But rather than attributing that action to any independent receptor substance, his idea was that the hormone must bind directly to the enzyme and allosterically alter its activity. You can notice that he had, in this scheme, no receptors and no G proteins. All of this was to come over the next 15 or so years due to the work on G proteins, as you heard, of Rodbell and Gilman, and the receptors by my laboratory. Again, as Chris mentioned, we discovered this large family of G protein coupled receptors, beginning with the beta adrenergic receptor, discovering its architectural similarity to and amino acid homology with the visual pigment rhodopsin. We then were able to ultimately not only isolate the receptors, but clone them and learn about their amino acid sequences. We ultimately were cloned about a dozen of these G protein coupled receptors, not just for adrenaline, but for other molecules like serotonin. They all had this highly conserved structure. And then the field expanded dramatically. We now know there are probably about 800 of these receptors, different receptors in humans. They regulate virtually all known physiological processes, and not just respond to hormones and drugs and neurotransmitters, but they also mediate our sense of certain tastes like bitter and sweet, also smell, also vision through rhodopsin and the color opsids. But again, as Chris mentioned, most notably from the view of clinical practice, the GPCRs, or G protein coupled receptors, are by far the largest family of drug targets. Several examples shown here, amounting to anywhere from a third to a half of all drug targets. And that is more than 700 drugs, 700 drugs. So I think there's a lesson here. I myself was never a clinical investigator. For that matter, for the most part, with a few exceptions, I've also not really been a translational investigator. I've been a basic, fundamental investigator. And nonetheless, the work has had very striking implications for the practice of clinical medicine. And I think it's worth remembering that even the most basic work can have important ramifications for therapeutics and clinical medicine. I think for young people trying to decide in which direction to go with their research, it's worth bearing in mind that all types of research, be it basic, clinical, or translational, can ultimately have dramatic ramifications for the practice of medicine. Now, as I mentioned, I think mentoring is very important, and I have mentored about 250 people in my laboratory, primarily postdoctoral fellows and several dozen graduate students, as well as, not counted in that 250, are many dozens of undergraduates. I currently have either 8 or 10 in my lab right now. So I have developed my own system of mentoring, and I wanted to share with you several of the conclusions that I've come to. The first is that you must tailor mentoring to each individual's needs. You need to be a bit of a psychologist here, and this takes experience, but every student or fellow is unique, and no two are alike. And so you have to learn what motivates one versus the other. And in fact, what you learn is what motivates one may turn another one off. So it's important to individualize your mentoring and realize that even for an individual trainee, their mentoring needs will evolve dramatically during the time that they are with you. You need to encourage focus. There are 4 keys to success in research, I always tell people. The first is focus. The second is focus. The third is focus. And the fourth you have to figure out for yourself. I realized the importance of teaching people to focus over the years by an experience that I would repeatedly have when I would travel. So let's say I would go out of town for a week for meetings, and then I would come back and I would try to get caught up with each of my trainees in terms of how the work had gone over the last week, and if they were what they had gotten out of the experiments we planned together before I left. And more often than I would like, I would find that very quickly a number of them had gotten off track. They had gotten a result which was perhaps a bit unexpected, and they were off chasing, going down some rabbit hole instead of staying focused on the goal at hand. And it reminded me of an experience that I had when I was in medical school. During the first 2 years, each student was required to have their own microscope. So there was a tradition at my medical school that senior or more senior students in the clinical years who didn't need the microscope anymore would pass it down to the freshmen. And so the microscopes we all had were really beat up and old, but they functioned, sort of. And so I remember sitting up in my dormitory room at Columbia overlooking the Hudson River, studying slides in the evening. And I would look away at the manual to see what I was supposed to be looking at. But by the time I put my head back, the stage had slipped and was out of focus. So I learned to put just the right amount of torque with my left hand on the fine-tuning knob so that the stage wouldn't slip. And I realized eventually that that's sort of one of the main things I did in the laboratory, was exert just enough torque on my individual trainees to keep them focused. Except, of course, on the occasion when it actually did make sense to pursue some unexpected finding. People often say, well, how do you know the difference? Well, again, there are no rules for that, but that's another thing that I think a skilled mentor can inculcate in his trainees, is a sense of when you pursue an unexpected finding and when you ignore it. Fan the flames of enthusiasm. Be enthusiastic about what you're doing. If you're not, find something else to do. When new people come to the lab, I always tell them, there are 2 conditions that are necessary but not sufficient for you to succeed here. First, you're enthusiastic about your project. Second, I'm enthusiastic about your project. This was brought home to me, the importance of this enthusiasm, which is so infectious, as we all know, by a story I was told by one of my trainees many, many years after he had left the laboratory and gone on to a very successful academic career. He told me that one night he was in the lab late and was talking with a couple of his mates in the laboratory, and he was telling them one of the things that kept him going is that he always felt that his project, when he talked to me, was by far, to me, as lab chief, the most important project in the lab. And that this always kept him, highly enthused about what he was doing. Well, then his mate said, well, wait a minute. I don't see how you drew that impression, because I've always been quite convinced my project was the most important one to Bob. And then the third guy piped up, well, what are you talking about? We all know that my project is the most important one to him. A few days later at lab meeting, they kind of jokingly confronted me on this. And I said, well, of course, you're all right. In the moment that I'm talking to you, your project is the most exciting and important one to me. So I think that enthusiasm from a mentor is very important. Teach trainees to build their careers around problems, not techniques. The reason for this is that simply that, as we all know, techniques have a certain half-life and are invariably either replaced or superseded by novel, newer technologies. And so if you tie your career to a particular technique and then go looking for problems, you're going to have a pretty short career. Rather, you should find your problem based on curiosity, what you're really interested in, and then figure out in the moment what are the most appropriate and powerful technologies to bring to bear on that problem. Promote risk-taking. Be bold. Take risks. Don't be afraid to fail. This is a subject that I could spend the entire hour on. Every successful scientist I've ever talked to, every really successful person I've ever talked to, will acknowledge the importance of dealing with failure, especially early in your career. I've heard innumerable talks by successful people from the world of business, law, entertainment, and to a person, they will tell you that learning to deal with failure and how to learn from failure is one of the most important things that you can do. And it was a lesson that was hard for me, but which I learned during my time at the NIH. I remember chatting with a senior scientist when I was in the depths of despair after a year of failure. And he said to me, Bob, do you know what percentage of experiments that an average scientist does are actually successful? I said, no, I don't. He said, well, it might be 1%. He said, but do you know what the success rate might be for a real world beater, maybe even a Nobel laureate? I said, no. He said, it could be as high as 2%. And that really impressed me, the fact that even for the best scientists, most of what they do is failure. And I remember there was a young scientist at Duke, not one who trained with me, but who I used to mentor over the years. And one year, late one afternoon during week between Christmas and New Year's, he dropped by my office and he said, hey, Bob, what are you doing? I explained I was fulfilling a ritual that I went through every year at that time and have for really 50 years now, which is going back over my last year of work, seeing what worked, what didn't work, what ideas turned out to be stupid, what ideas were basically stillborn. I didn't even pursue them because it became clear they made no sense, and which ones actually worked. And then based on that, I wrote up my goals for the next year, and a year later, I went through the same ritual. I said, how has your year been? He says, well, it's great. I said, well, what fraction of what you're doing would you say is working? He said, oh, I don't know. He says, just about everything, probably 90% or better. He said, how about you? I said, well, I'm right where I want to be. I would say maybe 15, 20%. He said, that's it? The great Lefkowitz is doing 1 in 5 project or less is working? I said, yeah. I said, some years it gets higher than that, but I'm not happy when it does. I said, and one year I actually got up close to 50%, and I realized I really needed to change course. I was really just sort of trying to harvest low-hanging fruit and doing derivative things. I said, so yeah, I don't like it when I'm much above a small percentage of things working. Anyway, he was dumbfounded by that, but he took it to heart. And over the years since, he's told me that it had a profound effect on him, and that he really had very much changed his approach. to work on more challenging things. And I can tell you from my own observation that his career has really flourished during that time. Model dogged persistence. I mean, there are projects that we've worked on in the lab, not for weeks or months or even a year or two, but for a decade or more. If it's a good problem and you continue to feel that it's tractable, then nothing better than persistence. Empower trainees. This is really one of the most important roles of a mentor. And there are two completely diametrically opposite ways of screwing this up. On the one, because you have to walk a very narrow line. On the one hand, you can micromanage the trainee, consistently tell them what to do, almost plan their experiments for them. But the risk in doing this is that when they finally succeed, they fail to develop the confidence in themselves that comes from an appropriate amount of independence. And what's always in the back of their mind is, well, you know, without Bob or whoever, I wouldn't have been able to do this. And so they lack that confidence to take on difficult and important problems. On the other hand, if you hang back too much, then they get nothing from you. They don't learn anything from you. They might as well not be in your laboratory. So learning how to tread that line is one of the most difficult things, especially since the line is different for each trainee. And as I said under point one, it's different in any given trainee as they proceed through their training. So again, it requires tremendous individualization. Emphasize storytelling. Learn to tell a good story. I actually learned this lesson, which is equally applicable in the clinical arena as in the research arena, when I was a medical student. I was a third-year student taking my very first clinical rotation in medicine, and I was doing it at Mount Sinai Hospital in New York. And on the very first day, I had this wonderful attending physician, Mortimer Beta. He was a pulmonologist, and I got to present the first case that I had admitted that day. And I made the presentation. He said it was very nice. We discussed the differential diagnosis. And then when I was done, he said, okay, Mr. Lefkowitz, now I'd like you to represent the case. You can't change any of the facts of the story. However, I want you to tell me a different story. Well, I was dumbfounded. I didn't know what to say. I couldn't do it. He went around the room, asked if anybody else could do it. Nobody could. So he showed us. He took the facts of the case, changed nothing, but he completely altered the emphasis and the order in which he presented findings, and told a rather different story, from which he derived a somewhat different differential diagnosis, ultimately leading, unlike mine, to what turned out to be the correct diagnosis. The lesson he was teaching was that data are not a story. A story is something you impose on the data with your own creativity. And in fact, that lesson applies perhaps even more dramatically to the scientific world. For example, when we're writing a grant or when we are writing a paper, and we take the different figure panels of the data that we have and we shuffle them around, trying to decide what tells the best story. Is this figure the first figure or the last figure? Or is it somewhere in the middle? So again, it's worth learning how to tell a good story because data themselves are not a story. Laugh and have fun. Try to see the humor in everyday situations. If you have the right attitude, it's all around you. I find that humor is a great prod to creativity, and I try to leaven my lab and other meetings with a generous amount. You know, when you think about it, there are some interesting analogies between humor and scientific discovery. In each case, for example, when you say something funny, often it's a matter of seeing a relationship between seemingly incongruous events and putting them together in a way that strikes people as funny. In other words, in the moment that you as a listener see the joke, in a sense, you're making a tiny little discovery about seeing a relationship between things that you might not have seen before. And of course, that is the basis of scientific discovery. is seeing relationships between seemingly unrelated things that others may perhaps have missed. So I think humor is very important. I use a lot of it in my lab meetings, and I find that the more funny things or humorous things that are noted, the more people relax, the more they're having a good time, and the more the creative process just seems to be embellished. But there is one caveat here, and that's that I'm quite convinced that the good Lord created two types of people: people who are funny and people who are not funny. Now, even if you're not funny, and there's no crime in that, even if you're not funny, of course, you can still appreciate humor. But here's the key thing. If you are not funny, don't try to be funny. We've all seen the grotesquerie of somebody who is inherently not a funny person trying to be funny. And there's nothing more cringe-inducing than a non-funny person trying to tell a joke or trying to be funny. So, piece of advice here, if you're not funny, that's fine, just don't try to be funny. Respect your own mentors. I'm so fortunate the two mentors I had at the NIH, Jesse Roth and Ira Paston, are both still professionally active. Jesse's 89 and Ira is 90, so they're wonderful role models for me. Few additional keys to success in science and probably many other things. Do lots of experiments. Among the trainees I've had who were the most successful, a fairly general characteristic has been that they did more experiments than the average trainee. Don't talk yourself out of experiments too easily. Even if you have numerous reasons why an experiment won't work, consider doing it anyway. Often, you'll be surprised at the result. And if you want serendipity to operate for you, you really need to do the experiments and let serendipity go to work. Be ambitious. You really need to want to solve the problem that you're working on. Try to view your work as play. Now, of course, all of us have days, including me, when you have to take care of certain things that, you'd rather be doing something else. But I've been so fortunate in my career that a good number of my days, I feel like I'm playing in a sandbox and just enjoying myself and loving the process of doing science. Be optimistic, and preparation, and more preparation. And I'll say a word more about that in just a minute. But whether it's preparing for my every 5-year reviews at Howard Hughes Medical Institute, or working on a grant or a paper or a talk, there's no substitute for preparation and more preparation. I want to say a few words here at the end about some of the remarkable experiences that I've had as a Nobel laureate. It really does change your life. And even before I was a Nobel laureate, I got to meet a number of amazing people. I think I mentioned that, at the NIH, the quality of the people who went into that program was remarkable. I mean, in my class alone, that is, those of us who graduated medical school in '66 and came to the NIH in '68 as Yellow Berets. In my class alone, there were 4 future Nobel laureates. In addition to myself, there was Mike Brown, Joe Goldstein, and Harold Varmus. And one other guy in that class you may have heard of who didn't win a Nobel Prize, Tony Fauci, shown in this picture. Tony and I are good friends. We met at the NIH more than 50 years ago. We still keep up with each other. This is a picture of me and Tony some years later in the 1990s when we served on the council of an organization called the Association of American Physicians together. Got to meet President Bush when I received the National Medal of Science from him in a White House ceremony. Of course, I got to meet the King of Sweden when I received the Nobel Prize. I got to meet President Obama. He was fulfilling a long-standing tradition, which is that the American, and this goes back, I think, to President Eisenhower, I was told. The tradition is that the President of the United States greets all the American laureates when in December they're on their way to Stockholm to receive their Nobel Prizes. He was wonderful, had this wonderful self-deprecating sense of humor. You may recall he had received the Nobel Prize during his first year in office, the Peace Prize. And it surprised a lot of people, including him. He said, "You know, I have one thing in common with you folks. Like you, I too am a Nobel laureate." He says, "But there is one key difference." He says, "You earned yours." I got to meet Coach Mike Shayesky the day after I received the call from Stockholm. I did not know him at the time, but he called me up. I'm an avid Duke basketball fan, have been for decades. And he said, "Bob, I really relate to what you've just accomplished." He says, "As you know, I brought Duke our very first NCAA sports championship. Now you've brought our university its very first Nobel Prize. So I'd like you to come down to Cameron Indoor Stadium. That's the gym where the team plays. And we want to honor you." And I did that a week later. You can see the fans in the background with their hands raised in the air. And they presented me with a jersey with the number 1. And it's quite an amazing experience to be down there. I could hear the students who sit closest to the court chanting something at me and pointing like this. I couldn't quite hear what they were saying. And then it finally came into focus. They were saying, "He's so smart. He's so smart." So at that point, I just put my hands up in the air and I just let the sound wash over me. And it was really an amazing experience. Got a big hug from the coach. A few years later, they tried to give my number away to President Obama, but you can see that it's a smaller jersey. And this is me in my office, surrounded by my 2 most prized professional possessions. On one side, my Duke jersey, which is hanging from the rafters in my office. On the other side, the Nobel Prize. One other amazing experience that I had, which is one that a number of Nobel laureates have, is that I was invited to throw out the first pitch at a baseball game. And we don't have a major league team in Durham, but we do have the Durham Bulls, which is a AAA farm club, a minor league team. And so I was invited the summer after I won the prize to throw out the first pitch and be honored at a Durham Bulls game. This shows me at the ceremony before the big event. From left to right, that's Wool E. Bull, the team mascot, myself, Victor Zhao, who at the time was the CEO and president of Duke Medical Center, and with the microphone, the long-term mayor, William E. Bell. Fortunately for me, they gave me plenty of advance notice. I had about 2 months to get ready for this. And it's a good thing that I had that. Because although I played a lot of ball when I was a young man, I found, to my horror, now age 70 at the time that was going on, and that was like almost 10 years ago, I basically couldn't throw a ball 60 feet with any accuracy. In fact, most of the time I couldn't throw it 60 feet. Every once in a while I did throw it 60 feet, and it was totally wild. And so I really set diligently, as I told you, preparation, and more preparation. I really started to work very hard with almost anybody who would work with me. One of the most helpful was my eldest granddaughter, Maya, who lives in San Francisco and pitched for her high school team. On a business trip out there, I stopped in and she worked with me all afternoon at a park, taught me a lot. Really, probably the single biggest increment in my performance came from her. And then every day I would play catch during lunch hour with anybody from the lab who would catch me. And then finally, the last week before the big event, I called up the Duke baseball coach, and he set me up with the Duke pitching coach, and he worked with me from a mound. So I would learn how to throw off a mound and not just on a flat, which is a very different thing. And so the big day arrives, and you only get one shot at this. I was not nervous. I mean, I felt quietly confident, despite the fact that the people in the lab kept sending me these video montages of things like, worst 10 pitches ever thrown, first pitches ever thrown, and people falling off the mound and knocking themselves out, one guy throwing the ball at 90 degrees, another guy hitting an umpire, etc. So now the big day arrives, and I want to show you what went on. It's a little hard to follow the ball but just watch where the catcher catches it. You can see the Jumbotron in the background as I walk. You can see the focus here. Big windup and the throw. Perfect strike if you saw that he catches it waist high. And then ensued one of the proudest moments of my life. The catcher comes out, puts the ball in my mitt, looks me in the eye and says, quote, "Doc, that was one hell of a throw." And it doesn't get, I have to tell you, much better than that. Here was another experience, shows me with two aging rockers, Peter Gabriel and Sting, if you happen to recognize them. And you may be saying to yourself, what in the world are Peter Gabriel and Sting doing kneeling in a show of seeming respect, even admiration and awe, in front of Dr. Lefkowitz? And the answer is, I'm not going to tell you. You're going to have to read my book, my memoir, if you'd like to know that and many other stories. Finally, let me conclude with this. Nobel laureates seem to have a sense of wanting to tell other people, give other people advice about how they too can win the Nobel Prize. Mike Brown has done a TED talk on this. Mike Bishop has written a book called How to Win the Nobel Prize. I'm sure there are others, but I have a somewhat different take on it. And it's encapsulated in this paper called Chocolate Consumption, Cognitive Function, and Nobel Laureates, which ironically appeared in the prestigious New England Journal of Medicine on October 10th, 2012, the day I received the call from Stockholm. I did not learn of this article, however, for about another year. Now, what it purports to show, perhaps tongue-in-cheek, is that if you make a plot of chocolate consumption on the x-axis, this is sort of kilograms per year per individual, versus the number of Nobel laureates per 10 million population in different countries. If you plot those data, and each data point is the flag of the country in question, you see there's an almost perfect correlation with an extremely high significance level. Only one outlier, Sweden, seems to be getting more prizes than they deserve based on chocolate consumption, suggesting that perhaps they're tilting the scales of justice here in their own favor. So why do I show you this? Well, absolutely true story, God's honest truth. I have been a chocoholic throughout my career, my adult life. But because of my coronary disease and my desire to eat as little saturated fat as possible and keep my weight at ideal level, I've always limited my chocolate consumption, and this is dark chocolate, usually 75% cocoa. I've limited my consumption to 2 small squares, maybe a couple of nights a week. But then, in the summer of 2012, I said to my wife, look, I'm almost 70 years old. My coronary disease seems to be doing okay. My weight is good. My cholesterol is good. Why do I limit myself to 2 times a week? Why shouldn't I eat it every day? And she said, you should. You deserve it. And so sometime in July or August of 2012, honest to God, I upped my chocolate consumption from 2 days a week to 7 days a week. 2 months later, I won the Nobel Prize. So what can we conclude from that? Well, first, there's obviously a dose response here, and there's a threshold, and I did not exceed that threshold until I went to daily consumption. But equally important is that it's obviously very rapidly acting, because within 2 months of upping the dose, I won the prize. So for those of you who might aspire in the direction of winning a Nobel Prize, let me suggest dark chocolate may be the way to go. So I'll conclude on that note, just to mention, as Chris said, I recently published this memoir, A Funny Thing Happened on the Way to Stockholm. If you've enjoyed any of my stories and advice, I think you'll get a kick out of the book. I have lots of material on serious subjects like mentoring, what Nobel Week was like, how I returned to Poland to find my roots, where my great-grandparents and grandparents came from, etc. But also lots of light and humorous stories. So I'll conclude on that note and stop sharing. And if there are any questions or comments, I'll be happy to entertain them.
Well, thank you so much. We have already a few questions, and I could spend an hour with you more on scientific ground to ask you for some thoughts on that. But let's start with the question from our Vice Chancellor for Research. What is the reason that GPCR lead to so many drug targets?
It's a very good question. I think the first thing is that they do play such a crucial role in physiology. I mean, as I mentioned, they basically regulate virtually every physiological process in humans, and so they stand ready. If you can manipulate their activities with drugs, you can manipulate almost any process. So that's for openers. The other thing is there seems to be something about this 7-membrane spanning domain arrangement and the sort of barrel-like structure that they make, which allows this, what we call the orthosteric site. The orthosteric site is the site where the endogenous ligand binds. That's where adrenaline binds to the adrenergic receptor, where serotonin binds to serotonin receptors, where histamine binds to histamine receptors, etc. There seems to be something about that site which is just very druggable. So anyway, those are a few thoughts on that.
So you have 600 of those receptors, and the question I'm sure was posed to you multiple times: how is that happening that the structure could be virtually, in a first approximation, overlay? What is thermodynamically or on a chemical side make them so unique?
You are correct, Chris, that I have been asked this question on multiple occasions, and on just as many occasions I've responded with total hand waving, and I keep waiting for some physical chemist or biophysicist to provide an explanation for what it is about this particular structure which has enabled it to be utilized in evolution for so many totally divergent purposes. But I do not have an answer to that question.
So that means there are still possibilities of getting Nobel Prize for GPCRs.
I would say so. If anybody can figure out what it is about this structure, and despite the huge explosion of structural biology and biophysical work on these receptors in the last 15 years, I still haven't seen a good explanation for that.
We have a question from Philip Kaiser. Fantastic talk, Dr. Lefkowitz. Your emphasis on early education in public school, you emphasize that. And do you support public schooling for elementary, high school educator over private schools in general? And whether we as a scientist should participate in this education program with high schoolers and earlier on?
I absolutely do. You know, when you think about the approach that in the United States and countries all over the world take to sports, I think we could learn a lesson. So let's take the Olympics. Every 4 years, we and most of the countries around the world send our very best athletes to the Olympics. And how is it that we have these very best athletes? Well, we begin very early on when kids are in grade school, plucking out the most talented kids and putting them into special schools or special programs, I should say, sports camps, etc. I think we should do something similar in education. I think in different scientific areas, etc., mathematics, physics. I think, one, we should be educating the kids in elementary school with simple concepts, but we should also be plucking out those who are particularly talented and putting them into special classes to nurture that. A perfect example of this is the Bronx High School of Science. We actually have a high school in Durham, which is patterned after the Bronx High School of Science, but rather than just drawing on a city, as we did in New York, the Bronx High School of Science, this is a statewide school, and it's a residence high school. It's called the North Carolina School for Science and Mathematics. but it's the same idea. You're admitted based on a competitive examination. But to give you an idea of what this can achieve is the Bronx High School of Science is, I don't know, somewhere between 75 and 80 years old. And I told you the only criterion for admission is scoring high on a competitive exam. I'm the 8th Nobel laureate to graduate from that high school. The other 7 were in physics. Think about that. If the Bronx High School of Science were a country and not a high school, there would only be something like 20 countries in the world that have more Nobel laureates. And this is one high school. I was giving a talk in Korea virtually about a week ago, and I got a question. In Korea, we've never had a Nobel Prize in a scientific discipline. Any suggestions? We'd love to have a Nobel laureate. And I spoke along these lines. So there are plenty of countries that have never had a Nobel laureate. And as I say, there are only 19 or 20 countries that have had more Nobel laureates than this one high school. So I think that's what can be done if you really try to pick out future stars early on.
Bob, you get a lot of praises here for the audience, and let me finish with one of my story. I wanted to assure you I'm still happily married, although you were instigating almost our divorce. As we collaborated on rhodopsin kinase, and I finished the prep very, very late in Christmas Eve. And so we survived that. So you don't need to worry anymore.
Can I tell that story, Chris?
Of course.
Yeah, okay. So we're going back to, I don't know when this was, probably in the 90s sometime. Chris is just finishing up his postdoc in Florida and getting ready to move to, where was it, Seattle?
Oregon.
Oregon. Okay. And we had just discovered an enzyme, which we called BARK, beta-adrenergic receptor kinase, which phosphorylated the receptors as the first step in what is now known as a universal mechanism for turning off the receptors, but also allowing them to signal through an alternate signal transducer called beta-arrestin. Anyway, contemporaneous with our work on this enzyme, Chris had been working, and who was your mentor in those days? I can't remember. Hargrave. Paul Hargrave was working on an analogous enzyme called rhodopsin kinase, which phosphorylated rhodopsin in a light-dependent way, much as our enzyme BARK phosphorylated the beta receptor in an adrenaline kind of way. So we had just cloned the gene for BARK, and it seemed to be the founding member based on amino acid sequence of a new subfamily of kinases. So we thought that rhodopsin kinase might be a member of the same family, since we had already shown that rhodopsin kinase in vitro could weakly phosphorylate the beta receptor, and the BARK could weakly phosphorylate rhodopsin. So I called Paul Hargrave and I said, we would love to get one of your preps of rhodopsin kinase so we could fragment it, get some sequence, and use that to design nucleotide probes to clone its gene. He said, well, the only guy around here who knows how to purify it is Chris Palczewski, and he's just leaving to move west. And this was right around Christmas time. He said, so if you can talk him into doing this prep, more power to you. But I mean, the poor guy is getting ready to move his family. It's not a good time as the holidays are coming. So I said, well, I'll take a shot at it. So I called Chris, who I didn't know at the time, and explained the problem to him. And he said, well, I'm leaving. And, he begged for mercy, but I didn't show him. And I just kept pounding away at him, saying, you got to help me out. Anyway, I can be pretty persuasive, I guess, and I talked him into it. But his poor wife was not happy about it, and I didn't blame her. The holidays were coming. They were supposed to move. Poor guy, but he stuck around, bless his soul. He did the prep. As you heard, he finished it up on Christmas Eve, and then he moved west, sent it to us. That's all we needed. We took that one prep, pristine, pure protein. We cleaved it up with cyanogen bromide. We got several peptide sequences, and we were able to clone the gene, cDNA. Sure enough, It looked very, very similar to GRK2. And so we now knew that there was a new protein family, a new kinase family. And without Chris doing that heroic preparation, well, wouldn't have happened.
Thank you, Bob, very much. Dean Stamos?
Thank you very much. Oh, wow. What a great talk. I know that we can't hear or see our attendees, but please join me in a round of applause for Nobel laureate, Dr. Robert Lefkowitz. We are so thankful you could share your morning with us today.
My pleasure indeed. I'm glad to do it.
What a truly inspirational, enjoyable, educational, and humorous talk. Really fantastic. On a personal level, I want to thank you for doing it, but I also want to thank you especially for your recommendations for dark chocolate intake. Absolutely. I love and I eat a lot of it, so I'm hoping there will be some ongoing benefit to my cognition.
Absolutely, I can guarantee it.
I only wish that you had also endorsed red wine as an accompaniment.
Well, that's fine. That's, shall we say, optional, but certainly approved.
Before we leave, I'd like to thank a few people for their support of this event. In addition to my esteemed colleagues and partners on this lecture, Dr. Abbott, Dr. Cooperman, Dr. Palczewski. Appreciation also goes to a few other people for their behind-the-scenes support in making today possible. First, William Alvarez and Kyle Good from UCI Media, who pulled off this Zoom presentation without a hitch. I want to also thank Zayana Aiden from the Center for Translational Vision Research. I want to thank Michelle Strombeck, Jessica Banfield, and Emily Welsh from School of Medicine Communications. And then last but not least, Emily Dawadoff and Rachel Correll from the School of Medicine Dean's Office. So once again, thanks everybody for attending. Thank you again, Dr. Lefkowitz, Dr. Palczewski, Dr. Cooperman, and Dr. Abbott for your excellent contributions. Have a wonderful day.
Thank you.